Good afternoon, ladies and gentlemen. I am Krishna Kalluri, alternate counselor of the Southern California local section of the American Chemical Society. It is my pleasure to introduce today's distinguished speaker, Professor Devin Peterson. Before I do, let me say a few words about the National Chemistry Week celebrated by the American Chemical Society. Check the ACS Matters October 14th, 2025 issue for a whole lot of information about the NCW, that is the National Chemistry Week. I will just quote a couple of sentences from that. Get ready to spice things up. This year's National Chemistry Week during October 19th to 25th explores the hidden life of spices. From the molecular makeup of cinnamon to the volatile compounds in chili peppers, NCW invites all of us to uncover the chemistry behind flavor, aroma, and tradition, and discover how chemistry connects to culture, cuisine, and curiosity. Now, let me introduce today's speaker. Professor Devin Peterson is a distinguished professor in the College of Food, Agriculture, and Environmental Sciences at the Ohio State University. He received the Presidential Early Career Award for Scientists and Engineers, PCASE, that's what it is called, Excellence in Flavor Science Award from the Flavor and Extract Manufacturers Association, IFT Stephen Change Award for Lipid and Flavor Science, and the Fred Kavli Innovations in Chemistry Lecture Award by the American Chemical Society. His research focuses on identifying and analyzing the chemical stimuli that contribute to the flavor of food, including gustation, that is taste, olfaction, that is smell, and somatosensation, pungency, irritation, and tactile. He is the founding director of the Flavor Research and Education Center and is the faculty director of the Ohio State University Foods for Health Theme Initiative. He has published more than 121 peer-reviewed scientific articles, and today's title on which he is going to talk about is Unraveling Flavor Chemistry, Sensory Experience, and Food Innovation. Now over to Professor Peterson for his seminar.
Thank you, Dr. Krishna, for the certainly that introduction and appreciate some context, you'd say, to some of my background. I'm going to talk again. I shortened my title, as you see here, but really maybe just give you a little bit of an insight into, how do you think about, applying the field of chemistry into something that affects our everyday life. That's really probably what got me interested in this space, is the idea of applying maybe science to something that is you'd say an important experience for humans and dictates a lot of things that I'll try to give you some appreciation for throughout the context of my slides. I will be so I am featured on the Chemistry Week next when they talk about spices, so some of that work will be from my lab, so there'll be a little redundancy of me if you tune in next week. But at least I can give you some broader context of the things we're interested in, and certainly some of the activities in my program, and certainly some of the things we try to have impact towards. So let me just quickly give you a little bit of an overview of me. And so I just moved my program to something called this interdisciplinary research facility called the Pelotonia Research Center. Actually, if you look at the very bottom corner here, this is where I'm sitting actually currently, the bottom right. And I'm sort of, you'd say, a little bit of maybe where does flavor research and maybe even food science fit into, some of the aspects of our, how science can be having more role and more impact on our daily life. And I would actually try to say health and wellness is certainly one of them. And so this building is certainly one way that Ohio State is trying to obviously spur and catalyze, interdisciplinary type research. And so instead of being siloed in our individual departments, taking, a few people from across different departments and putting them in this building that just allows a little bit more crosstalk and hopefully collaboration in a way that is meaningful. And so I'm proud to be part of what I would say, how can agriculture provide more leadership and thinking about, I would say, food and health, and some aspects of this that I'll try to at least provide some thoughts on from my lens. So, these aren't new quotes. Almost a decade ago, there's this quote that came out, and it basically says here that poor diets are responsible for more of the, burden of ill health, the largest risk factor, if you will, to ill health than sexually transmitted diseases, drugs, alcohol, and tobacco combined. And that's a pretty staggering statistic. So in other words, it's saying that really the other way of looking at this is food could be doing so much more, okay, to maintain a quality of life, as well as, to have, you would say, longer, more active, healthy lives. And what I would like to just paraphrase this a little bit, and when we say poor diets, really what we're referring to is dietary patterns. And so if I look at the graph on the right, what you'll see is if you just look at epidemiological data, these longitudinal studies, they will basically have, in this case, by this sort of process, have sort of indicated that basically these poor dietary patterns contribute to about 22% of deaths if we look at the global sort of, you'd say, diet and sort of death, you'd say, connections. And what you're basically showing you here is that we don't consume enough whole grain. We consume too much sodium, for example, in a way that, again, results in a lower quality of life and a shorter life. And so, it's interesting, if I was to take the same graph and do high income North America, obviously where we are, it only goes down to about 18%. And so, one of the things to think about here is that I guess that certainly what we eat is very influential in our overall quality of life, and more thinking about prevention than treatment. So certainly a lot of research in the United States is focused on cancer research and other aspects, right? Heart disease and drug discoveries that really are part of those, solutions and prolonging your life, et cetera. What this is, I would say, agriculture in the United States and broadly is more of a commodity. And there's, I think, a really big opportunity for us to think about food beyond just a commodity, and how do we elevate its quality, if you will, in a way that just has a lot of potential for, you'd say, translational impact, to humanity. And so, again, this idea, and I want to be careful. I think the food, if you went back 100 years, certainly the food today there's been tremendous advancements. You go into a grocery store and you can get fruit from all over the world. And, so it kind of comes back to a couple things. And what is, at least in part, driving this, like, why are we eating poor dietary patterns? And so part of that, what I would tell you at least, is a major factor that influences what we eat is we eat for pleasure, okay, not typically for health. And humans among the animal kingdom are a little bit unique from a variety of reasons, but a big part of what we're going to consume is baked into pleasure. I'm sure many of you, when you celebrate, you do that with food you enjoy. And so, certainly everybody has, I think, more understanding of this, and certainly I think gravitates towards trying to eat healthier. But regardless, if you look at the statistics and sort of consumption behaviors, I would, a big component of what is driving dietary patterns is certainly going to be, I'm picking this because I want to enjoy my meal. And so, that then gets into this idea of, why is healthy eating so difficult? And I think that, as I was sort of mentioning before, I think a large part of this, because agriculture is based on a commodity. It was affordable, abundant. And those are good things, actually, right? You want your food supply to be economical and reaches more people and less of a barrier. But it also comes sometimes at a cost, in that we're not really designing food to be, if you will, healthy choices, to be more aligned with human behavior and ingested behavior. And even if we think about sustainability, right, and you think about, the goal is, if you look at, agricultural practices and thinking about, global warming and other aspects of the world, how do we maybe align, our agriculture industry to be more sustainable? Well, the goal of that is, you could put on the package it's more sustainable, but that doesn't sell, unfortunately. What sells is what I want to eat. And so you have to kind of align consumer behavior, I would say, with these larger initiatives, and just an understanding of what is ingested behavior. And just an example here, Bezos Foundations, right? are, the whole idea of meat analogs and sort of maybe removing some removing some animal to get more plant. Well, the question of that again is how do we maybe again do this in an affordable, in a way that also meets consumer demands? And it's just a very challenging question to take plant materials and make them consume or make them mimic meat. And so I'm going to talk a little about that, right? So why is this so difficult? It is a big analytical challenge to try to understand what I'm perceiving. And I'll try to, go through a few examples of that in a way that, hopefully piques your curiosity as a scientist and perhaps, ideas of how you might think about, future careers. So this article was just released last week. It's from a lab out of Purdue, and it's basically without me getting into a big discussion on this. I guess what I'm trying to point out here is that food choice is complicated. There's a lot of inputs. Certainly things like culture and socioeconomic and income and food deserts, but without question, still a major player is going to be acceptance. It's hard for me to imagine a world where healthy eating doesn't mean enjoying your food. I just don't know that I see that aligned very well, and I think there's challenges with that. And so anyways, with regard to just to sort of point out, this is very complicated, but I would argue that understanding these sort of key consumer traits of these products is still has relevance, and in this case, relevance to health. NIH used to fund flavor research 50 years ago, and then somehow said this is a consumer issue. This has nothing to do with health. And I couldn't disagree more. I do think understanding how to make choices more compelling to consumers is a way to get them to eat healthier, and understanding a lot about just ingested behavior. So when I see things like precision nutrition and ingested behavior in a title sentence, I like to see that because I do think there's a better need for us to understand, these attributes in a way that is more enabling, I would say, for designing more sustainable, healthier food supply. So just very, very quickly, this is mentioned that I, sort of direct this sort of, public-private partnership consortium. I'm a big believer in, as a researcher, to have public-private partnerships. I actually think there's a different opportunity for science when you're trying to solve a problem. I think it's easier to be creative. I think there's a value that comes from this within the scientific, you would say, platforms, right? And sort of discovery processes. And so, having my ear to the ground, listening to companies as they try to maybe reduce salt and or, promote other aspects, how can I maybe have some understanding of that at the same time grow our fundamental understanding of what is important for ingested behavior in a way that can elevate healthier choices, actually, in a way that, I think can provide some outcome. So as was mentioned, at the very beginning, of my introduction. So what is sort of interesting about flavor research and discovery is, for the last 5 decades, really aroma has been considered to be the primary component of flavor. It's the most important, is what I think a lot of people would potentially indicate to you. I don't agree with that. I think it's an important part. I don't know that I think it's the most important part. So I don't think, it's not that I would ignore it, but I think it contributes to diversity. And I think reason why we came to this, because you could smell thousands of different, perceptions. For example, gustation, taste might be 5, right? 5 different, sweet, sour, salty, bitter, umami. Right. And so because of this, our mind says, well, this is more simplistic. This is more complicated. The complicated's got to be more important. And I would say, I think I don't think is as true as people thought it to be. Let me put it another way. The other part is somatosensory. And so, or I should say somatosensations is to keep things maybe more consistent along my terms. But what this is, again, is, so these are 3 sensory systems in the oral cavity. And they, olfaction has millions of receptors, same with gustation, and somatosensory also. You have these millions of chemical antennae in your oral cavity that, as you're drinking a beverage or you're eating something, are responding to those chemistries very particularly and putting feedback to your brain. And these are perceived in different parts of the brain, are then integrated into a synthetic precept. And so, you can't think of this as individual molecules. You have to think about it as an integration of stimuli, okay, in that regard. And so what I would also just from my lens, and sorry, I don't know how I lost my references on here, but nonetheless, so what I would, I guess, basically give you some context is if we've looked at flavor research for again the last 5 decades, what I would say is that well, you know, most research has sort of gone to a reductionist approach and said that only things that are agonists are important to perception. In other words, they break things apart and look for where your nose smells, for example, or you taste, quantify it, right? And then make those up in a way to say this is what is active. If I look at receptor biology, actually, most of the what's called is these, the G protein coupled receptors, most of them are actually for perception in your body. Many are for other aspects. You have the same receptors in your oral cavity throughout your body, actually. And what is kind of fun is actually like the taste receptors are in my stomach. They're linked to my pancreas, but they don't function for taste. They're doing other things. They're sending other biochemical, are the part of biochemistry, I guess, and sort of ways that our body's responding to its environment. But what I would say again is that, historically, again, we've always focused on what's an agonist. In other words, I'm looking for individual molecules that have perception. If I'm a receptor biologist, what I would say is that is certainly a known component of what triggers receptors, but is not the only thing. It's much more complicated. You can have things that are antagonists. You can have things that, allosteric or, generally modulators, things that might enhance and or suppress, right, activity in ways that creates more complexity to understanding stimuli. So if I'm trying to understand wheat, and I just mentioned lack consumption of whole grain is one of the major contributors to poor dietary patterns. And if I went to a breeder in Kansas where they have a big breeding program, big genomics program, so they can really understand wheat quality, right? Uh, from an agronomic perspective. So it's all about yield because the farmer is paid on yield. But if I was to say, I want to make sure that as you develop wheat, it's tasty, right? It is flavorful. They would have no idea what to look for. They just don't have an understanding of what is driving, they would say, consumer patterns, right? In a way that is important and influential. Okay. And so, why do we make it so difficult? It's because we're treating it like a commodity. Okay. And we don't really have a great understanding from a scientific lens. We're, really evolving these materials more for agronomy traits than human traits. And I think that's a big, I would say, disconnect in a way that has implications. What I would, maybe just really quickly point out is there's some work, again, was published in Cell, and basically what, they're looking at here is, okay, we have 10 different odor receptors. I'm going to say in human, you have about 300, right, that are functional. And what the point of this data is saying is that when I look at volatiles and I take a certain amount of volatiles and I start looking at my receptors, um, the volatiles behave more as antagonists than they do as agonists. And that's interesting because if I only look for if I only look for an agonist, again, an individual molecule that has perception, I'm missing part of the story. I'm missing part of the how that maybe, if I'm lighting up millions of different receptors and different patterns, and I'm only looking at agonist activity, but not really also considering antagonist activity, that I miss the way that odor is shaped and an understanding that now is important for perception. So, if we go forward a little bit, and I'm going to get into some data after the slide and try to give you some examples of things we've looked at and maybe ideas around, again, what are the molecules you perceive? So if you made something from scratch at home in your kitchen, or you go out to a restaurant you enjoy, or fast food, what is it that, right, when I'm eating, what are these molecules that are ultimately triggering perception in a way that is, impacting my behavior? And so as I sort of show you here on the left, a lot of analytical methodologies actually have sort of are a hybrid between humans and instruments. So, again, most work being done on aroma, on olfaction. And from that lens, you know, um, GCMS would be probably the preferred tool, because the GC has the highest resolving power, for a variety of reasons, and can really pull apart complex mixtures. And then I put a mass spec on it, and it helps me then to think about chemical information and understanding all the plethora of different molecules that could be there. From a identification point of view, we're probably somewhere around 12,000 volatile molecules have been reported, okay, in food and beverages. So quite a diversity, right? When you think about flavor molecules, they can range in chemical class and it's just a tremendous diversity, right, in the type of molecules that exist and that are in food, in nature, and things that impact our daily life. But where I was kind of going with this again is we will then take, oops, I gotta fix my mouse here. But let me move to my trackpad and maybe less of this going back and forth. So what I would say is that, so what we end up doing is you'll take something like you're interested in, and you'll run it through a GC. You go through sample preparation, isolate out a chemical mixture, typically of the volatile fraction, inject it, get separation, and actually smell at the end of the column, because the instrument will see a lot of chemical signals, but it has no idea what you smell or don't smell. And so this is a big part of how we sort of evolved the field, is that people will actually smell the end of the effluent and say, oh, I smell nutty here, I smell musty, or I smell peachy. If I take butter flavor, which is a highly desirable ingredient from a culinary chef, and I put butter aroma, I isolate it out, I make it volatile, I think about some ways of doing that, and I inject it, I will smell things like cabbage, baby vomit, earthy, peachy, but when I put them together, give me butter. So you don't smell, a lot of times, again, it's the integration of all these different individual parts that have a higher, you would say, integration into what we call the desirability of butter. It's not one molecule that's butter, okay, for that matter. Now, in margarine, that's a different issue. We'll all stop there. But I think what I'm trying to show you here is just some of the complexities of perception, that you don't have this, you have these very interesting different molecules that often don't represent the flavor at all, but yet when put together, create something very unique, okay, in that regard. So on the left, again, is kind of this idea where we have, in this case, how we would understand what are some of the molecules that we perceive. Once we identify things by smelling, okay, what's called olfactometry, we will then identify them, quantify them. Often some of these can be part per billion, part per trillion, very complex to get good quantitative data, right? And this is not trivial. And we'll go back and figure out what all these, these antagonists are. Sorry, agonists are. Let me get my terms correct for you. Agonists, right? Things that have direct perception. And then, from here, basically do and compare it to what's called a sensory threshold value. So that's where 50% of the population can detect it. And anything that is, above threshold is considered to be important and contributing to that perception. So my lab, probably about, I don't know, 12 or so years ago, we do a variety of things in the lab, but what I would say is we start to think about and adopting from the field of metabolomics a little bit, basically the idea of saying, we have really good analytical chemistry and analytical methodologies today to see molecular signatures. Can we now couple that with statistics and have a different way of thinking about flavor discovery? And so in this case, what we do is we create really good chemical prints, and we look at an attribute of interest like flavor, and then try to then develop models to predict what is related to that attribute of interest. And so that's a big difference, right? Because one is looking only at in this case agonists. The other is looking at what is predictive based on stats, right? Based on my ability to map variation. And so those are two different things. And what that allows you to do is to maybe think, like I showed you for on receptors, is something that's not, are all only things that are agonists, only things that have smells on their own. Is that all that matters? Okay, in the bigger picture, is that practically what's important? Okay, so this is the question, right? The other thing would be is if I said, okay, I can take butter and I can break it into subcomponents and understand those subcomponents. But to actually understand liking is a little bit more complicated. That's an integrated component to a human. These methods, I can also look at what is driving maybe liking or disliking, which is different, right? When I'm smelling individual molecules, it's hard for me to say, does this contribute to liking or disliking? If I smell mushroom, I was doing my butter analysis, I might say that's disliking. No, it's an important part for butter, right? So it's very hard for us to take these individual, you would say, sub components and predict things that relate to liking, and in a very generalized way. I think sometimes that works. I have something like nutty, okay, that might contribute to something that's important for roasted hazelnuts, but it still doesn't always, maybe there's still some limitations to that. And so just to give you some context, but the idea is, can we use more, I would say, these, we'll call them non-targeted, untargeted is probably the more common term that's used today. And can we use these methodologies, okay, for understanding? So let me go forward here, and we'll talk about coffee. Okay, and coffee is something that touches a billion people a day. I mean, it's hard to think of products that do that. There's only a few, right, that touch that many people every day. And one of the things that is rapidly changing right now for coffee is climate change is really putting an impact on the coffee industry. And why? It's because they're just getting more insect damage and changes in rainfall, among other things. They're just not surviving. They're not able to, you would say, have the same yields and have the same opportunities as previous. And this is a beautiful picture from Hawaii. And one of the varieties that is grown in Hawaii is called Typica. And as of a couple of years ago, I think about 50% of the crop has now been lost to insects. They just can't keep up. And yes, they try to use insecticides and this and less ideal. At the end of the day, of course, making sure residues are the right levels. But what I'm also trying to say is the coffee industry is rapidly shifting right now, and I think you're going to find in 5 or so years it's going to change. And so, what does that mean? So these you would say bushes, right, that are growing these you would say coffee beans, right? If I talk to geneticists, they know, again, agronomic traits. They know how to breed them for maybe insect resistance or drought resistance, agronomic things, but they have no idea how to breed them for flavor. So historically, we've had a lot of time. You can grow a lot of different things, and you kind of, cross things that are maybe desirable with things that are less, that are more economic. But the challenge with this is it's not always, as effective in time, and you have a climate that's rapidly changing, that they have a lot of different varietals right now that they think can provide climate resistance, but they don't know that they're going to meet the quality standards in a way that's beneficial for business. And let me explain, okay, why does that matter? You're like, who cares? I mean, grow something and then people drink it. Perhaps, but let me give you a little bit of how the world works today. So currently, the coffee industry, if I look at this globally, okay, probably from a market capitalization, the coffee industry could be benchmarked anywhere like up to $200 billion. It is a big business, right? Again, lots of people consume coffee, daily. And so, it's not hard for the math to add up quick. And one of the things that's important for coffee and how it's monetized actually is how its flavor is. So currently, as a farmer is growing this in Guatemala, depending on the flavor that he's growing, is how the beans are going to be monetized, right? Not just yield. It is actually very much based on this because there's a certain quality the consumer expects. Okay, so one of the things that we would say is, okay, so what is important, right? What are the molecules in coffee that are important for its perception? And why does that matter? Well, if we understand more of these molecular foundations, that opens up breeding opportunities and selection basis, right? For breeding tools, as well as even just how do I cultivate the coffee and even roast it, among other things, to make sure that it's going to give the quality that is needed for its value. And so these things translate all the way to the farmer. So if it's sub-quality coffee, they're going to get much lower, I would say, profit margin from their crops than if it's a higher quality. So these things have, I would say, implications across the whole supply chain, okay, in that regard. And so if I look at coffee again, I'm going to show you a little bit of how we think about discovery and maybe some some understanding of, again, what is flavor chemistry. This is more this well-called non-targeted or untargeted, kind of strategy, where again, the goal is to use variation to our advantage and statistical methodology to help us understand molecular compositions that are providing impacts, right, on, in this case, the flavor in a way that is related to its monetary value. And so I'll show you in a second, but what one of my prior PhD students do is we source the coffee globally, work with the professional sort of organization in the coffee world, we used humans, like what I'll show you in a second, called cupping, which is this way that they will valorize coffee beans. And then basically took chemical prints to these and said, can we understand from the chemical compositions what is actually influential, okay, for the overall quality in a way that provides now knowledge and science to, in this case, hopefully help with, elevating the crop quality that, again, translates broadly. And so what I will tell you, again, because coffee is 200 billion, I can guarantee you there's been a lot of research that's been done on coffee. But it's largely been done on the smell. If anybody drinks a cup of coffee, the smell is quite signature, right? It's important. It is an important aspect, with very distinguishing. You walk into a coffee house and you can smell the coffee, and it has characteristic components to it. Well, for this particular work, I really wanted the student to think about also the non-volatiles. Non-volatiles you don't smell. Okay, you need to have volatility for them to reach the olfactory region. They have to have vapor pressure, and so I guess when I say non-volatile, I'm now eliminating them from, in this case, olfaction. Okay, from a practical sense. And so I had her really focus on not GCMS, which is where most people would go and say, hey, I want to understand what's driving coffee quality. I'm going to look at the GCMS because I'm really going to now use this for more volatile material. And we're going to create these chemical profiles of all these samples, basically create models, right, statistical models to say what's, driving, what among these compositions is predictive of things we like or dislike. Often then, I'll talk about this in a second, but many of the compounds are unknowns, actually, and that's very common for us. We identify probably on a yearly basis easily 12 novel molecules every year, no problem. Things that have never been reported in nature before. So we have to have a strategy for that. How do we identify unknowns? And then obviously, quantify things, identify things. And then actually a big part of what we do is not just create predictive models. So what you could look at the first, say, sequence of steps, maybe through to the bottom here, is more about what called hypothesis seeking is a common term, right? Show me what I don't understand, to hypothesis testing, demonstrating causality. And so I'm going to go through this process with you a little bit and just give you a little bit of this feedback, okay, on how this works. Okay, so what we can do is, if you've ever watched what's called a Q grader or a cupper, these are people that are trained. They have extensive training to evaluate the flavor attributes of coffee. They do what's called this infusion method. They put the grounds of coffee in some water at certain temperatures. Everything's very controlled, and they this slurping, like it's just loud, and they're foaming this stuff in their mouth and going through a whole range of things. And basically rate 10 different attributes that relate, if you will, to the experience of the flavor. And based on that, they assign it a cup score, and that cup score monetizes things globally, a commodity, or you would say an agriculture commodity, in the billions of dollar range. A very, something that touches, again, our everyday life. So what we did was we sought out to say, can we use our discovery platforms to do a little bit more understanding? I understand aroma that's already there. I understand some of the bitterness, I understand some of the sourness. So that's kind of already understood. Is there a different way for me to think about compositions as they relate to monetary value? And so what Sachaya did, the PhD student of this, is that we sourced these 18 different coffees. We had them cupped. I don't show the error bar here. I'll show it in another graph. But basically, this is an average of 5, these really, you would say, highly trained cuppers, and ranging from, 80 or 70, maybe 3 to 88-ish. And what is important here, one, is that we see a range in cup scores, right? So now I can say, can I chemically print these samples well enough that I can understand what is affecting their monetary value? right? That's the goal. And so again, if things are subspecially below 80, they are given a very low value from a product point of view, and the farmers paid less. And what I understand from the coffee community is that actually most coffee globally is subspecialty. So a lot of room for improvement potentially, assuming the market is there for specialty coffee, but potentially allowing one, better enjoyment of the products that you're consuming, two, quality of life, I would argue in that regard, and two, just more monetary value, right? That allows, you'd say, farmers and throughout the whole supply chain just have better opportunities that way. So I'm gonna show you quickly here. This is some data from a while ago, but it was some of the first data that we had worked on. And what I want to show you here is this is, again, you take the 18 samples and we take chemical prints of them. And when we take chemical prints, we certainly spend quite a bit of time making sure we have high quality chemical data, right? A big part of, you ever talk about big data, it's, I would say high quality big data is a better way. You don't want noise, right? So we spent a lot of time working on our instrumental methods and just creating, you would say, enough, if you will, depth of sensitivity and robustness of data that enables true discovery, okay, in this regard. And what I will say to you here is we have maybe, I think it's just over 1,000, we'll call them features, things that are thought to be distinct chemical compounds in those samples. And in this case, we create what's called a PLS regression. And what the nice thing here is this, just with, I don't have any GC data here at all. I have LC data, highly weighted towards non-volatiles, certainly going to be some volatiles in here. But what it's basically saying is without even the GC, the most common tool for flavor analysis, I can highly predict, right? Cup scores. I have, robust, I would say, predictable models. Okay. They're not overfitted, without me getting into the metrics of say the stats on it. I'm just going to say we have high quality model, and it can have a very low error rate. So, the cup scores out of 100, it can sort of predict it just from a practicality, like about a unit, right? 1 unit. So it's pretty good. Just at looking at the chemical position, saying, if I understand these, this is what, the model I can use for prediction. So we can take this data and say, okay, with the chemical positions, they're able to match the variation cup scores well. What we can also do then is plot the data on something called an S-plot. This depends on, I'm going to scale my data and other ways of thinking about, sort of data analysis. But in this case, we're going to do what's called Pareto scaling. And what we can do is we can say, okay, I'm going to take all these dots. So over here, I should say, sorry, is a scores plot. These individual dots represent a different coffee sample, maybe in duplicate, right? Or depending on how much we ran our samples, biological samples here. Over here, each of these dots represent an individual chemical in the coffee. So what you can do is we can develop these highly predictive models, and then we can plot the chemical data and say what is predictive, okay? And so what we can then look at here is, okay, so these red dots up here are positively correlated. So you have correlation on the y-axis and you have covariance on the x-axis. The more something ranges, the more correlated it is, the more it's going to influence my model, right? The more it can, in this case, understand the variation in my y variable. And so you could look at, it's called a variable importance and projection, a VIP score. But regardless of that, let me not dwell on this too much. What I can tell you, so this dot right up here is a compound, and it's the most predictive compound in my model to predict cup scores. Again, something that relates to monetary value. We go look at this compound with our LCMS, and it is an unknown. We can't find a spectra in any database that says it's this. That's interesting, right? So, okay, at this point, at least we could say literature has never reported it, or at least to the best of our ability from our library searching, okay? So the most predictive of something that affects the quality of that coffee is never been reported. That's kind of interesting, okay? Conversely, I can go down to the bottom and look at these, what's called negative correlated. So again, positively correlated as they're going up, cup scores go up. Negatively correlated as they go up, cup scores go down. Okay, in other words, less value, less quality. So then the question would be, is this, I'm going to say, well, let's start with the positives. And this positively correlated compound, right, is an unknown. So what do we do? Okay, we can do MSMS and certainly has some ideas of that, but it's really hard to get a decent structure, I would argue, or a more confirmed structure without NMR. And so what we do is then we basically go forward. My lab has a whole lab dedicated to preparative LCMS, where we will take something like coffee, though that may not look like a very rich TIC. If you've done LCMS, this has thousands of molecules in it. And the question is now, can I take the one that is predictive that I showed you, isolate it out, and purify it, of course, for what? So I can now do further, you'd say, structural elucidation, get my NMR, understand now, bond atoms, how they're connected, and of course, provide a standard for me, right? I can synthesize these molecules later. That's a strategy depending on how complicated they are. It might be easier for me actually to pull them from food. And so, or from coffee in this case. And so what we do is we'll take this and run it down multiple dimensions of LCMS, try to figure out what's the best sort of, if you will, separation, and often go through sometimes 3, sometimes 4, sometimes 5 dimensions of separation to ultimately end up with single molecules that are at least 90% pure in a way that gives us more assurance, okay, this molecule is the one that I'm actually perceiving. It's not the contaminant, okay, which is always a challenge. So this is a massive bottleneck. And I would say from my lab, I have to say this is probably one of the things that probably burns the students out the most is this. It may look straightforward. It is a bottleneck and it's a trivial, but getting enough quantity to do NMR and further testing on is certainly something we're continuing trying to evolve and work with in our in our local pipeline. So what is this molecule? Okay, this so-called molecule, the most predictive of cup score. Well, okay, I can tell from the mass spec that it was this chlorogenic, you'd say derivative of chlorogenic acids are well, reported in this case. Oh gosh, let me go back to my other mouse. So this part of the moiety is well reported in coffee. What has never been reported actually is this sort of methyl butanoate, adduct to the quinic acid. And so this particular one never been reported in Nature. Of course, we do NMR, NMS, and whatnot, characterize the molecule and everything else. But so what we do is, one of the first things I have the students do is, okay, this molecule is very predictive of something, again, that's important for quality of life and we'll say just enjoyment, but also monetary value, as I keep saying. In the most like coffee, the highest cup score coffee, it is 2.3 ppm, okay, milligrams per liter of coffee. We make it up at that range. We do this in a very controlled way. And actually, by what calls IRB approvals and human testing, we actually evaluate these things in a way that is still acceptable. And so we take this molecule, we put it at the level that, again, is in the most highest quality coffee, put the pH that is of the coffee, just to sort of make sure we don't have any pH effects. And there's really no flavor activity. There's nothing. Okay, so one could say, you just have a correlated molecule that's not causative, right? You have something that's inversely correlated, or in this case, not related, just happens to be related, or maybe from other aspects, but not related to the quality. Okay, so that could be possible. Or could it be, as I mentioned, something else is now contributing in a way that's not common to the current paradigm in flavor? Flavor, again, being agonist, I have to perceive it. Because I didn't perceive this molecule, most flavor chemists globally would have just thrown it out. It doesn't matter. I don't perceive it at the level that's in coffee. Therefore, it is not influencing quality. Okay. So here's this most predictive molecule. Again, novel to nature, never been reported before. So what do we do? Okay, the next thing we would do is go into what I call hypothesis testing mode. And so what is kind of fun here is that we would basically take these molecules and go back and test them blind. The people who are evaluating them don't know anything about them. They just know their coffee, right? So they don't know what they're trying. They're all 3-digit coded cups. And basically what we'll do is we'll take, in this case, if I can get my mouse here, we'll take a subspecialty coffee, okay? I'll show you that data in a second. We know the level of that compound, this predictive compound, is about, we'll say 1.3 ppm. In the most like coffee, as I just showed you, it was about 3.2 ppm. So we add the difference, right, to this lower quality cup and have people try these all 3 samples and say, give me your evaluation, drink them, tell me if they're different. Okay. And so what is kind of fun about that is, again, the cuppers don't know what they're trying. And I remember when I sent this to the company who does this cupping, first they open up these glass bottles, because what we did is we had ppm levels of these compounds, like a milligram or two in these glass bottles that they fill coffee into to help, if you will, then create the treatment sample. And they looked at it and said, oh, there's nothing in here. I'm like, you just can't see it. It's that low, right? It's coded, and it's like, ppm levels is very not simple to deal with, is what I'm trying to say. But we, of course, check everything to make sure when we do it this way, we measure them and we can show, yes, quantitatively, they are adjusting as we are predicting, right? That when we recombine things, they're doing what we expect them to do to create that new level. And so what we're going to do now is I'm going to show you basically, okay, this coffee, plus these coffees, right? These treatments, these controlled treatments, and just see what happened when the cuppers went and did this, okay, in this regard. And so what I should have put on this graph is actually, these are all the treatments down here, right? And so if it says control, it just means it's the same sample plus that compound, okay? And so what is kind of fun about this is that if you look at this coffee, here's a subspecialty, this control, it's just below 80. Okay, this 80 point is where you would have subspecialty, the value drops quite a bit. It'd be more what I'll call industrial grade coffee. So we add this compound 1, that's the most predictive, the one I showed you here before, and it increases the cup score when we add it just under 2 ppm, like 5 points. We're probably not as familiar with, this kind of coffee chemistry, if you will, but that's massive. That is enough to change its monetary value, like a whole category. And what is sort of really fun about that is, again, this molecule by itself has no perception. So flavor is what? Okay, this is I'm going to end here because I just take some time to talk through some ideas. But what am I saying to you ultimately? I'm saying to you that flavor is complicated, a lot of receptors, and what I'm ultimately have to understand when I think about what is these things that really influence the quality of these products, if you will, consumption behavior, ingestion behavior, is that I have patterns of molecules that don't operate independently. So, okay, this one most predictive, the second most predictive is actually a very similar molecule, but it's the quinide. So what happens during roasting is you just get basically these dehydration product. The hydroxyl binds with obviously the carboxyl group up here and forms a lactone ring. And so you have this ring structure. This happens by me taking the bean and roasting it. It's losing moisture, and it goes through some dehydration reactions. And here's this now, all the infinite number of molecules in coffee. The top 2 predictive ones are very similar structures. Okay. The second one only increases the cup score when it's a quinide, slightly different structure. Think about the receptors, the chemical structure, even stereoisomers will react differently. So very tuned to very unique, very particular structures, right? That are very influential now, right? In this very low level. And the other thing I'll just say too is, okay, these top 2 predictive compounds, right, are influencing this overall quality, flavorless again. They have no flavor. Now, if I drink them on themselves, it doesn't matter. But what it's basically saying is the molecules in coffee need these other molecules to then do something that is now creating a higher value, right? A different sort of quality. What is also fun about this, if you take all these 4 compounds that are predictive, 3 that are statistical, 1 that was not, at least at the end that we analyzed, when I add them all together, I don't get additive. Often people think about more like this is better, this one's better, so I put them together, I'm gonna get better, even more better. And it's not, it's it's an average. And so again, talking about these receptors and this sort of antennas in the oral cavity, and ultimately how this chemical composition is not about individuals, it's about a complex pattern of individual molecules that we need to try to understand, right, in this regard. So I'm going to actually stop there and see if there's questions. But I would just say we've done these things like on hazelnuts. They're trying to domesticate hazelnuts in the US. We have an American hazelnut that can grow in much of the Midwest, different than the one in Europe. And the idea for this is, I've worked on this again, is if you want to domesticate something here, let's not make it so you have to coat it in chocolate or sugar or salt to eat it, right? This doesn't make sense. There's much flavor diversity in these materials, but when they're bred for yield and disease resistance, right? Kind of intertwined. And can we think about ways, again, that would make you want to eat nuts or tomatoes or whole grain? And these are the kind of things that we try to unpack. I just showed you this fun example of coffee, but I certainly have worked on a whole range of things. And then if you are part of Chemistry Week next week, you'll see my lab get highlighted in a way that we look at how we might think about reducing salt, right, in foods and still not have what's called a salt penalty. Are there ways that we can enhance the perception of salt so you can add less and still be, yes, this is the right salt level? And therefore, again, remember sodium being one of the largest contributors to poor dietary patterns. Okay, with that, let me just park it and see if there's any questions that you would like to ask.
Hi there, can you hear me?
Can you hear me?
I can now, Dr. Keller. You were muted.
Yeah. So Lucky Morales asks, how does C4 compare to C1? Have you increased the concentration of C1 relative to C2 and
Yeah.
mix up final bar?
So I didn't explain this very well. And sometimes when you do this, you're talking high level. I'm happy if you looked at that publication, there's certainly be a lot more details in there if you're, at all curious. What actually C1 and C2 are different molecules, right? So C1 is this, in this case, this chlorogenic acid with this, methyl butyrate ester. It's the exact same molecule, but over here, it's a dehydration, okay, product of it. Again, so this actually is endogenous in green beans. This happens when during roasting. It doesn't all go, but you have some of it being formed during roasting. And so just showing you these two individuals, this actually is an unknown. The student didn't identify it just based on how far she got. And so we tried some of these molecules, even though we didn't know what they are. We know we have enough molecular signature to even evaluate them from a sensory. So they're different C1, C2, C3, and C4, all individual molecules that are distinct. And then the mixture of all four of them together at the end.
Understood. Can you hear me or?
I can.
Awesome, great. Um, yeah, so I can see, yeah, it seems like it is agonistic in nature. There's some kind of competitiveness. I'm wondering if when you mix, because on that final bar, it looks like you mixed all of those compounds, but I'm wondering if you mix compound 1 and 2 in different ratios is where that sort of competitiveness can be maximized.
Yeah. So what this data is, I think what you're seeing is what it's implying in is, say this went up 3 points and this went up 5 points. I add them together, I don't get 8 points or even more. I get an average, right? I'm getting a competition between them. That's what the data is saying. Like the second the C2 only raised it, in this case, 2 points, so it was just less effective, right? On the doses. And the doses we're using are based on the endogenous levels of the coffee samples, right? I'm relatively careful when we give people molecules in food. We don't give them anything that's not what would be in nature. So often, if I went and surveyed a lot of coffees, I might get more variation. Okay, that molecule, and I could maybe extend the range a little more. But a little bit of this is more proof of concept. And my point, I suppose, if I have anything here, is that non-volatiles, in particular, things that are modulating, that are not agonists on their own, are influential. And I can show this with many products, right? In the end of the day, as we've gone and done, our research. And that's kind of also part of I would say what's new, what have we brought to the table in regards to understanding stimuli?
Understood. Thank you. Also
You're
very
welcome.
impressed seeing the p-values attached with the graph. I don't often see them, but thank you.
We try to do better with that. Sometimes you get off graphs by mistake, but the pub should have all the details.
Devin, I have one question.
Please.
Actually, supposing you compare the agonists or antagonists, whatever it is, of a child with an adult, the child seems it does not like anything that is really spicy, but a good number of adults, like me, for example, we like things that are very spicy. Is there a
Yeah.
difference in the agonist and antagonism sensors when you are a child and when you grow up?
So what I can say, a couple of just general things. Culture has a big impact on your preferences, right? It's you can grow to adapt, especially aroma. You get used to things. You start to, if you will, there's a lot of receptor work on talking about, these kind of, you would say, experiences and how multiple experiences, push you to liking and other aspects, right? And or at least shape some of those preference maps. As you age, generally your perceptions get less. So especially in elderly populations, one of the issues is even in, for example, cancer can also result in, diminishing perceptions. As you age and in your very elder years, you start losing sensitivity, and it actually is certainly related and understood to affect your food ingestion. So you just stop eating and you don't eat as much, actually, in a way that's not great for your long-term care or health. And so, so I would say there are genetic differences without question. People talk about super tasters. I think that's more academic than I would say practical from a, my own lens. But I will say that a couple things I didn't show, but we've done some really fun stuff, like even in beer, like some examples of can I understand beer liking? And what you'll find is that it's really hard to take consumers and understand how they might differentiate by genetics or culture or these other variables that are important for their preferences. And so one of the things that we're working on right now is using machine learning and other tools, right, to help us pull out from a population cohorts that help us now to connect chemistry to them. So these molecules might affect 30% of the population, or elderly, or young people, or these cultures, right, in a way that is more, encompassing of understanding human behavior. But that is a very much more challenging area. When you look at most of this data, they just take a consumer panel that has been trained, right, or a trained panel, I should say, not a consumer panel, a trained panel. Then the other spectrum is consumers, which are untrained. You're sort of recruiting perhaps a cohort of people, but I think where I'm trying to go is, yes, there's ranges, right, that vary by many factors.
Okay. So, and there's a question from Wellington Oliveira. I would like to know about the safety of using a compound obtained from a preparative LC system in sensory analysis. Are there any treatments for the solvents used in these compounds to ensure that no additives from the solvents will be consumed by the panelists?
Yeah. So, so what I would say too is that we're very, very careful of the solvents we use. And in fact, typically use methanol and or ethanol for most for extraction. I will tell you, if you're in the food space, you can use hexanes, you can use methylene chloride. There are lots of solvents that are actually approved by FDA that are deemed safe, right? If these are high purity solvents and you're extracting out, organic matter, and then the goal of that is you're using volatile solvents, right? So you can flash them off. That is, at least I would say, within the food space, considered to be absolutely okay. For my lab, I'm very careful on the type of solvents we use and making sure the residue is well below anything you get in nature. I can tell you, if you go take food at home right now, and I was to analyze hexane, I will find it. It is everywhere. So we have to be really careful when we talk about this. And I'm not suggesting that it means we don't take risk or we don't care about risk. But I can guarantee you, if I take food, the average level of hexane in any agriculture commodity is probably half a ppm-ish. It just exists in the world. We live in a bubble. So the stuff is everywhere. And so what I guess what I'm trying to say is we absolutely certainly go through what's called IRB approvals, and again are very careful about the molecules we're identifying and understand them and from nature, et cetera, and are very even the environment we are isolating these in are very controlled environments, right? They are not like in a standard lab. They are separated. The glassware, all the stuff that touches things are all, you would say, very much regulated in a way that would be, I guess you'd say, congruent with safety protocols. And again, certainly do not give doses. Most of the times when we're trying things, it's like you take a mouthful. Most people will eat, I don't know, like many mouthful, right? When they're doing something, or a cup of coffee is many sips. But we never consume like a whole cup of coffee's worth. We would consume a sip worth, right? Or 2 sips worth. So it's a little bit of us making sure the dose is always typically, if you looked at toxicology is part of the poison. We can look at also the chemical motifs and run them through toxicology type, you'd say, programming to get some cues of things. But mostly, again, being very cautious of residues and just making sure there's nothing that would ever be even close to what you normally consume in your regular life.
From my own experience as a chromatographer, I see that the solvents that we use for chromatography, they are absolutely very pure, and their MS or any other technique does not show any impurity or any material at all in them. Because if they have any impurity, a lot of our LC analysis, LCMS analysis are going to be screwed up.
So you're correct. I mean, many of these are part per billion and low, right?
Yes.
I would still say we're very careful in that what we use, and kind of the residues, right, are, and you think about we're now dosing this at part per million typically, when most of these molecules are at a part per million. So even if they had 8 part per million or something, now you're talking about, subpart per billion, right, and per
Yes.
quadrillion. And so
Mhm.
not that means we don't care, but I can tell you, if I take beer or I take scotch or I take food you're eating, there are many molecules in there, right? And I guess the goal of this is to make sure that, again, we're not doing anything that would be above and beyond your typical life.
There's another question in the chat from Nancy Cordoba. From your perspective, what's the best approach to translate these findings back to green coffee or cultivation factors, given that roasting acts as a strong modulator on the final chemistry?
Sorry, if it's Nancy that I know, it's actually a prior postdoc of mine. So what I would say is, so what happens, right, is if you look at this molecule that's mostly endogenous to the green bean, I think there's also part of where does it come from, the green bean is the question could be asked. But when we've measured this, it's been measured basically from the standard cupping. So it's like a medium roast type protocol. What certainly is apparent, though, is you will get direct transformations, right, between the endogenous molecule all the way through to the quinide form. The quinide form is still active, it's just less active, right, at the end of the day. And so, I guess what I would say is that I don't know that we've really parameterized, but certainly more heat, more drying would result in more quinide formation without question. And so it goes back to, a little bit of balancing the different attributes you're looking for, along with some of these modulators. And we're doing a lot more work now, I would say currently in the lab, of how these modulators work, right, within the food context. So what are the molecules they're actually modulating? So that we have better understandings of, I think, some of the questions you're getting at to better optimize, products like coffee.
If there are no further questions, it is my pleasant duty to thank Professor Devin Peterson to have accepted our invitation and present a very interesting seminar on coffee and its aroma and flavor. Thank you very much for all of you for attending. And the recording is going to be on our website, www.scalex.org. So anyone interested can actually go to that site and listen to the recording. Thank you very much.
Thank you as well.